Hello everyone. For today we have some notes on crossing over and sex-linked traits. As always, if you need to pause the video, go back, go forward, whatever, do as you need to get your notes filled in. Okay, so crossing over. We briefly talked about crossing over when we talked about mitosis and meiosis. At its core, crossing over is just an exchange of chromosome segments between non-sister chromatids. So let's say that red is from mom and blue is from dad. It blends together, and then you get one that's part mom, part dad, another that's part dad, part mom. This is why if you have siblings, you're not exactly the same, because your parents' chromosomes can cross over in prophase 1 of meiosis so that it's a new kind of trait, a new blended chromosome. So again, it happens during prophase 1 of meiosis, and if you remember, meiosis increases genetic diversity by creating sex cells, whereas mitosis creates genetically identical daughter cells. So where does it occur? Where does this occur? Well, genetic swapping occurs between paired homologous, remember back to that word, the prefix homo means same, homologous chromosomes meaning like two chromosome number 1s or two chromosome number 2s. So the same chromosome, but one from mom, one from dad. So this is the egg and the sperm, or sex cells. New genetic material has been acquired or gathered from the other homologue. So maybe mom's chromosome 1 mixes with dad's chromosome 1, and now that mom's chromosome that was originally from mom that may inherit to the kid also is part dad. Now, genes that have a tendency to remain together during crossing over are said to be linked. What this means, kind of picture format. If you have two chromosomes together, right, from mom, from dad, let's say A, B, C, D, and they're all paired up. E and F is paired up. When they get together, if it's really close, like this A and C is really close, but it's not close to these ones on the tail end. The ones on the tail end are more likely to cross over and be exchanged than the ones that are really close together. So at its core, if it's close together on the chromosome, it's likely to move with it. If it's far apart on the chromosome, it's likely to be swapped. Now, we call that process gene mapping. So this is tracking crossing over, helps determines where these genes are located. Because we can look, if these traits don't seem to separate when meiosis occurs, then probably they're located closely together on a chromosome. So genes that are far apart have a greater chance of crossing over. Genes that are closer together are less likely, have a less like are less likely to cross over. Wow, just winning with the English here. Now, genes that stay together are said to be linked. One gene can be identified as a marker, and that can infer the presence of the other gene. So if we know the gene for eye color is at the very end of a chromosome, and we see that eye color seems to be linked with hair color in some way, then we're going to guess that they're close together. But like your eyebrow color and your hair color are controlled by two different genes. Often they're the same because they're close. They can be different, but often they are the same. Now, sex-linked traits. Calico cats are the best example because calico cats can only be female, and this is due to genetics. We call this sex-linked inheritance because it is linked to the sex chromosomes. So the cat's fur color is linked to alleles on the X chromosome. In order for a cat to have two X chromosomes, it's going to be biologically female. Two X chromosomes means it has two colors. It has both the allele for black fur and the allele for orange fur, which are on the X chromosomes. So sex-linked inheritance means that a gene or multiple genes are carried on one or more of the sex chromosomes. Chromosomes that are not X or Y, which are sex chromosomes, are called autosomal chromosomes. So that's the first 22 pairs or 44 chromosomes. Humans have 46, the last being the X's and the Y's. So sex chromosomes versus autosomal chromosomes. Sex-linked inheritance. Biological females have XX. Biological males are XY. The Y has very few genes on it that it can carry, maybe about 87, whereas the X can carry over 2,000 different genes. A lot of information is found on this X chromosome. And if you remember, it all kind of goes back to this karyotyping lab we did. Here's a karyotype of a XX, here's a karyotype of an X, and there's the little Y. In comparison, the Y chromosome is significantly smaller than the X because it carries less genetic information. So some genes for a disorder or disease are located on one of these chromosomes. If the gene is linked to a Y chromosome, if you're biologically female, you can't have it. Why? Because you don't have a Y chromosome. So it will only be on people who have a Y chromosome. We did talk about other conditions where you could be like XXY or XYY. So there's always exceptions to the rule. Trust me. Now, if the gene is linked to the X chromosome and is recessive, women, most likely biological women, will not express the disorder. Why? Because they have two X chromosomes. So if they get one good copy and one bad copy, they can rely on the good copy. Whereas if you only have an X and a Y, if you get one bad copy, you get whatever that gene is. So males are more often affected by sex-linked disorder because, like I said, they only have one X chromosome, which makes them way more likely to carry on whatever that recessive trait is. If you have two X chromosomes, you're less likely to receive those disorders. Now, females can be carriers because they can carry the gene without expressing it. If you have two X's, one is good and one is bad, you will show the good trait, but if you were to have a kid and pass that X on, you could pass on that bad trait, the bad X or whatever. It's sometimes not bad, sometimes just like color blindness, which is inconvenient. Affected males never pass the disease to their sons, because if you are male and you have a kid, you have an X and a Y. In order for that kid to be male, it has to get a Y chromosome, meaning it wouldn't get that damaged X. But affected males will always pass the defective X to their daughters because they, in order to have a daughter, the male has to pass the X. Because XX, daughter, XY, male. I said daughter and male. Anyway, you understand what I mean. Now, female carriers can pass the defective X to half of their kids. Half of their sons will have it, half of their daughters will have it, but their daughters will be carrier. All this stuff on this thing will make more sense once we actually try to do the Punnett square. Okay? Sorry about it. It gets a little weird. So how do you solve sex-linked problems? Well, you first determine if the trait is dominant or recessive, capital O. You set up a Punnett square where you say XX for female, XY for the male. You assign the alleles to the X's only. So if you notice on here, it's X big R, and X little r, Y. The Y doesn't get an extra letter to it because it's simply saying to code for testosterone. The Y chromosome has no allele. So this question says, if red eyes are dominant and sex-linked, show the cross between a homozygous, two of the same, red-eyed female, and a white-eyed male. He only gets one little r because he only has one X. This is very similar to the blood typing problems where the blood typing alleles are carried on the eyes. Well, this is just the same as homozygous dominant, two big R's, and heterozygous for just something that doesn't carry it. He only has one letter. That's it. And he's going to show that trait because he only has one X. So XX with two big R's passes them along. This female can only give X big R to all her kids. This male can only give a little r to his daughters and a Y to his sons, meaning this cross, a white-eyed male and a red-eyed female, all the sons will be red-eyed, all the females will be red-eyed, but they will carry the recessive gene, so they could pass it on. But the phenotypes is all the offsprings will have red eyes. So on yours, try this one out. So hemophilia is sex-linked, where X big H gives normal blood clotting and is dominant to the hemophilia allele X little h. So X little h gives you hemophilia. A woman with normal blood clotting whose father had hemophilia, we could also call her a carrier. A normal man whose father had hemophilia. Now that doesn't matter because he only has one X. What is the probability that a mating between these two individuals will produce a child, regardless of sex, that has hemophilia? So you set up your square. She's a carrier, meaning she's X big H, X little h. He's normal, he's X big H, Y. So set this up where you have X big H, X little h, X big H, and Y, and see if you can figure out the genotypes and the phenotypes. You do not have to put the square on your notes, just put the genotypes and phenotypes. So find a little scratch piece of paper, give it a try. If you get it wrong, that's okay. But set it up with this parent and see what happens to the kid. The question is, will any of them get hemophilia? All right. Well, good luck with that question. Thank you for getting your notes done, and I will see you guys later.